Good morning, everyone. Welcome to another EU OpenScreen webinar. This is a series of webinars running throughout the year on a monthly basis, and they are specifically dedicated to young scientists and researchers. I'm Kathi Skopelitou, the Scientific Coordinator in EU OpenScreen, and today I have the pleasure to welcome another great scientist, Professor Fernanda Borges, who is an Associate Professor with habilitation at the Department of Chemistry and Biochemistry in the Sciences Faculty of Porto University, one of EU OpenScreen partner sites, and coordinator of the Drug Discovery Group at the Chemistry Research Center of the same university. Fernanda's current research is focused on the design and development of new chemical entities to be used in the prevention therapy of neurodegenerative diseases, liver or infectious diseases. She has published more than 350 articles and 30 book chapters, and she is also the co-founder of the startup Mitotag and principal inventor in 6 patents. Today, Fernanda is going to talk about mitochondriotropic antioxidants and how they can be considered as druggable targets. Before I give the speech to Fernanda, I would like to remind you that this webinar is being recorded, and at the end of the webinar, we'll be running a live Q&A session, so you can ask your question using the Q&A box at the bottom of your screen, or raise your hand to ask your question directly to our speaker. Fernanda, I think we are ready for your presentation. Thank you for being here today.
Thank you very much for the kind invitation, and good morning to everyone. I try during my presentation not to speak too much about chemistry, because I'm not chemistry by formation. I'm a pharmacist. But how we can input our knowledge to advance in science, mainly with some drawbacks that we still have nowadays. Remember that for neurodegenerative disease, even nowadays, we have no effective pharmacological therapy. We have only in therapy symptomatic drugs that alleviate the symptoms of neurodegenerative disease. We don't have drugs for liver disease. And we are, by now, after the pandemics, with a pressing need of new antibiotics. So we have a lot to do, mainly the young research that has the opportunity to solve these problems and try to have new solutions for these problems that are growing up as we have a higher longevity in our countries, and these problems are also escalating. So we need to be hand by hand between the different experts in the field, from chemistry to biology, in order to quickly find a solution. I think that is our obligation for the society. So today I choose, because it was a running invitation, so I choose to tell you a history. How mitochondriotropic antioxidants have been developed, how our group have been engaged in this research, and what can they do in the next future to take part of this need of our society. So I will share my screen. I'll try to make a long story shorter and try to explain you that we are dealing with, when we talk with in my mitochondria, We are dealing with complexity, and we try to show, and other researchers, of course, that it can be druggable targets. So the first step is to tell something about our group. We are located in Porto, the city of Porto, and we are a group that has 10 young researchers with PhD, 16 PhD students, and also master and undergraduate students. As usual in medicinal chemistry, we have different backgrounds from pharmacists, chemists, biologists, and biochemistry field, because we need this expertise in order to move on our endeavor. As Katya said, we are focused mainly in diseases that have unmet they have no therapy and need that mainly academy, as most of the pharmaceutical industry give up from this to find new solutions for this disease. And also we have a field of nanomedicine, because sometimes we need to escape to some problems like permeability, toxicity, and formulate the best compounds in nano carriers. Usually we are dedicated to discover new chemical entities. The easy way is to focus in classic targets, but we are very focused in the discovery of new chemical entities for new targets, and contribute with some chemistry to the target validation. As usual, we are also playing in trying to try to understand the mechanism of these new chemical entities, bioavailability, and also toxicity studies. We have a starting point that is the scaffolds that are chosen to modify, and usually we choose our scaffolds in nature, as you see here. These are scaffolds that are found in nature that are used as starting point in order to be modified and have the adequate biological activity to the target that is under study. This is our workflow. We work like pharmaceutical industry in rational drug design, and we have two ways to approach, or a target-based drug discovery, or a phenotype-based drug discovery. The way to move on till the drug candidate selection depends on the design, if it is based in a target or is a phenotype. If it is a phenotype-based drug discovery, we need, after the lead discovery process, we need to check the target in order to play in the optimization of our molecules. And we start also some preclinical studies. This is a cartoon of our way to work. We have sometimes in synthesis, in order to produce the libraries that are needed to discover the lead compounds. We have also some facilities that now are improved with this joint venture with OPENSCREEN for screening, in order to check the target and produce the structure activity relationships that are needed to move on and to understand what we need to modify chemically in the compounds. Sometimes we also add another parameter that is the pulpity. We evaluate for some compounds in order to understand their activity, some physicochemical properties, and also we are involved in the evaluation of the toxicity. After collecting this data, we move for lead optimization, where we tailored our compounds in order to improve the potency, selectivity, and drug-like properties. Meanwhile, we need to discover the pharmacophore, and all these procedures are supported also by molecular modeling. So this is more or less, in summary, what we are engaged in our group. As mentioned by Kathy, we are dedicated to neurodegenerative disease, and at present, in the most prevalent neurodegenerative disease that are Alzheimer's, Parkinson's, and also ALS. These are the most prevalent in our society, and the diseases that need urgently of a therapy, mainly a disease-modifying one. To summarize some topics on this research that has a long time on our group, we discover new chemical entities for classical targets. These are two examples. We have been engaged with these enzymes that are marked here in green, that are enzymes that regulate the levels of the neurotransmitters that are very low in this disease. And always we try to reduce the toxicity of compounds and improve the drug-like properties. But if you are interested, you can read some of these articles, the most recent articles that we have already published. The second question that arises, that is, why till now we don't have pharmacological therapy for this disease? And we think that could be that these diseases are very complex, are multifactorial. And in that way, we think that the paradigm of the pharmaceutical industry, that is one disease, one target, one drug, is not working in this field. So we move also for the new one, paradigm that is developing multi-target ligands. So we try that for the disease to develop a drug that can touch several targets at the same time. So this is also a difficult task, because we need to block targets that are in the same signaling pathway and with a molecule that don't have off-target problems in the other targets that are not modulated by this chemical entity. This is also some works that have been already published last year that are related with the problem of the new approach, the multi-target approach. And you see that is here in red, that we try to touch two targets at the same time, two targets that are related in the signaling pathway. We try to improve from our molecules the drug-like properties, and sometimes we also have the aim to repurpose our libraries for other targets. And this is some examples that you can, if interested, you can read how we can do that. Also, we, as I tell you before, we try to develop some nano careers in order to solve some problems of the most effective compounds. And you see that we are working with polymeric nanoparticles that are carriers that are very interesting when we want to address some compounds, some drugs to the brain. That is very difficult local to target the compounds. So coming back for our topic, and also talking about neurodegenerative disease, we start, and the other scientists in the world, to check if mitochondria could be a druggable target for this neurodegenerative disease. And if it is a druggable target, how can we modulate our candidates in order that they have an effective effect on mitochondria? In fact, if you read the recent, more recent literature about the topic, you can see that this disease share the same target, that is oxidative stress. And when we talk about oxidative stress, or nowadays redox stress, we can try to map the sources of this oxidative stress. At the top, we have mitochondria, but we have other factors that contribute for this oxidative stress. And our thought and the other research was, can we manage or lower the levels of this oxidative stress problem and minimize or prevent some problems that are related with this disease. So we try in first beginning of this story to study a little bit of mitochondria. When we saw this challenge, at the first time, we saw that it will be very difficult, because mitochondria is the organelles, are the organelles that are relevant to maintain homeostasis, to maintain bioenergetic, biosynthesis, and a lot of signaling pathways, and they are responsible for our health. But at the same time, when they are dysfunctional, they contribute for the disease, and mainly for the oxidative stress-related disease and mitochondrial disease. So this task was very complicated. So the studies on mitochondria dysfunction escalated in the last years. We saw that inside of mitochondria, we have a complexity of targets that can be changed when the mitochondria is dysfunctional, and can contribute for the oxidative stress events. We have, for instance, the part of a respiratory chain the increment of the levels of reactive oxygen specials inside. We have also alterations in mitochondrial DNA, decrease on ATP levels, the changing of buffering of calcium, and so on. So the challenge, as you see, is very, very, is a big one. But we know, and a lot of studies have been published in last years, that besides neurodegenerative disease, the same problem, mitochondrial dysfunction, has been associated to cancer and also metabolic disease like diabetes and some liver disease. So this is why in last part of this century, modulating mitochondrial dysfunction emerged, in fact, as an attractive therapeutic strategy and stimulated drug discovery and development projects. The first thought is about antioxidant therapy. We thought, and the other research, that if we have antioxidants that can work in preventing or minimizing oxidative stress, it could be a valuable approach to minimize the events that are associated with mitochondria dysfunction. This antioxidant therapy could be, for instance, used as preventive, as an adjuvant therapy, or a combined therapy, if it works, of course. We check, and other colleagues that are in field, what happens to the clinical trials with several antioxidants. And we check that some antioxidants either modify synthetically or endogenous antioxidants like coenzyme Q, phytochemical modulators, some supplements, lipoic acids have been submitted to clinical trials. But still now, we have no positive outcomes of these clinical trials. Some have been some have been not completed, and for those that are completed, we have no reports. In fact, we observed that in general, with these antioxidants, we have a valid in vitro data in and preclinical studies, but we have no benefits in human clinical trials. Although for this disease, the medicine recommends to take some dietary antioxidants, lower calorie intake, or physical exercise, we saw that when we have the antioxidants, endogenous, dietary, or modified chemically, we have some problems. We have that they have, we notice that they have allow systemic viability, a rapid excretion, and an extensive metabolism. When we talk of antioxidants from diets, we can look for this molecule that was studied. Long studies have been done with resveratrol, some clinical trials have been performed with resveratrol in order to prove that is a good dietary supplement or as the drug, and no, till now, no positive outcomes were obtained. We think that the problem is with, again, with bioavailability, excretion, sorry, and metabolism, but even more in difficulties in establish the human dose, the dose that is good for playing the clinical trials is very difficult uh, to have till now. We don't have uh, known antioxidants proved from our diet that prove that it's good to be a supplement for minimize or preventing some oxidative stress related events. Steve, and now we come back also again to mitochondria. The challenging for these antioxidants is very, very high because the organelle that is responsible for nivellate the reactive oxidative species is mitochondria. And in fact, it is an attractive target, but is a very tricky one for drug discovery. Because if you remember from your formation, I don't know your background, mitochondria dysfunction have a lot of targets. And the question is, can these dietary antioxidants target mitochondria? And one of these targets, what is the best target to choose? And after choosing the target, is this target druggable? Remember also that mitochondria is for us that are working in the health area, is also a biosensor for drug-induced toxicity in preclinical development. And I hope that you feel that to develop a drug that has no drug-induced toxicity and can target one of these targets, it's a very daunting challenge. So this is the landscape of antioxidants and mitochondria. It is very, very challenging. We have a lot of targets here, and we don't know how to modulate our molecules in order that we can touch the targets and have benefits for this process. So we can look after the disappointment of these clinical trials on antioxidants, we can look mitochondria as a multi-target organelle. And in a simple way, we can look to change the bioenergetics or regulate the bioenergetics of mitochondria or act on the dynamic part of mitochondria. It is an easy task? No, because sometimes when we touch the bioenergetics, sometimes we modify the dynamics of the mitochondria. But we like, as researchers, challenge, and so we need to check if we can do something with this target. In fact, the first researchers in the world that tried to solve the problem were Scholarship that unfortunately passed away this month, and McMurphy in two different institutions that try to develop the so-called mitochondriotropic antioxidants. Scholarship discovered that this cation that you have here, the triphenylphosphonin, can penetrate the membrane of the cells and also penetrate in mitochondria and change bioenergetic and biosynthesis and some signaling pathways. So after this knowledge, they decided that this cation could be a good carrier to put some antioxidants inside of mitochondria. And in fact, they developed two mitochondrial antioxidants. You see that in different parts of the world, they are very similar. It is rounded in red, the core of the antioxidant. In this, the scholarship choose an antioxidant of the plants, plastoquinone, and Mike Murphy chose an endogenous antioxidant, the backbone of coenzyme Q. After they link the carrier through an alkyl chain in order to put the antioxidant inside of mitochondria. This was a long way, but these two antioxidants gave us some clues because although they have a small therapeutic window because they have some toxicity, they are very potent, and so they enter in clinical trials for several diseases. Although some of them failed, this is an important step in the history of mitochondriotropic antioxidants. After these discoveries, some researchers around the world try to develop themselves other mitochondriotropic antioxidants also based or inspired in the endogenous antioxidant system. We have here one based on vitamin E, vitamin C, and also in the enzymes that are regulating oxidative stress, the levels of reactive oxygen species. These mitochondriotropic antioxidants, although we can have some reference in the literature, they have not moved on for clinical trials. Also, other authors try to use the same technology in order to include inside of the mitochondria some probes like tempo that can catch reactive oxygen species and lower the levels of these species inside of mitochondria and contributing for the maintenance of the organelle during the health states. And this is where we come also. We talk with Scholarship and Murphy, and we try to use as scaffold, not endogenous antioxidants, but dietary antioxidants, because they have the drawbacks that I explained to you before. And so we decide, why not to develop, also inspired in the technology of Scholashev and Murphy, to put these antioxidants inside of mitochondria. In this work, we need experts in mitochondria, of course, and we have the participation along more than 10 years of Dr. Paulo Oliveira, that is in Center of Neuroscience in a city close to Porto in Coimbra, that work and is an expert in mitochondrial toxicology, and this is for us a very, very good collaboration. We decide together to develop a career to put these two dietary antioxidants in mitochondria. The antioxidants are caffeic acid and gallic acid. These are present in fruits, wine, coffee, and so on. They take part of our diet. But if you look for these physical-chemical parameters that are related with gallic acid, you see that they are very hydrosoluble, and they have another drawback, that is the presence of a carboxylic acid that is a drawback to their transport along the membranes. So we decide, it was a long road to put caffeic acid inside of mitochondria, modifying chemically and put a code, a zip code that could transport this caffeic acid to the mitochondria. Along this work, we check if this antioxidant caffeic acid can work on the mitochondria by itself, but it is, we have negative results. It can work on mitochondria because it has no physical chemical properties to penetrate in mitochondria. So, first of all, we tried to develop the lead compound. This work was published in 2012, and it is a work that was done with Mike Murphy in order to check if we have the lead compound inside of mitochondria. Yes, it was positive, it can come in, but the efficacy is not very good, and so we start to modify the compound in order to have robust structure activity relationships. This is the schema of the synthetic pathways, and we synthesize a lot, a small library of this of these modifications, but it was ruled out by antioxidant essays because we must not lose the antioxidant properties by the redox properties, the lipophilicity, and also the evaluation of toxicity at cell level and mitochondrial level. At the end, we have a lot of mitochondrial assays in order to check what the compounds done to mitochondria, and also if they don't change mitochondria. After this long road where we modified the phenolic pattern, we modified the linker, we modified the spacer, maintaining the same cationic carrier. In 2017, we were able to patent the strategy and publish the first work on this development of new mitochondriotropic antioxidants based on dietary antioxidants. Till now, we have two drug candidates that are here in blue. The candidate in blue, the candidate that is based on gallic acid. In red, the candidate that is based on caffeic acid. We check that we have no deleterious effect on mitochondria bioenergetics, on mitochondria morphology, and these compounds were tested in several cell lines and don't present toxicity. But they must have some good, this is very good, of course, but we need to prove that they have effects on the oxidative stress when the mitochondria is dysfunctional. And they proved to lower the mitochondria reactive oxygen levels. They prevent iron and lipid induced damage. They improved the levels of antioxidant endogenous systems. This is a hormetic-like effect. They improved the cell's resistance to oxidative stress. They activated some signaling pathways that are dependent or regulate the antioxidant defense, and they show neuroprotective effects and also BBB permeability. So at the end, after a long, more than 5 years, we have some good candidates to move on. And meanwhile, we have published some works after patent, either in the part of the fine-tuning of these compounds in order that they improve the efficacy and preventive performance of oxidative stress. We also publish some works with development of bioanalytical methods in order to evaluate their physical-chemical properties, and some are still ongoing to be published. After have these candidates that have a good performance, we need more robust preclinical data. So we establish collaborations in order that we can move on in with the animal models and prove the concept in order to move on for clinical essays. With also with Paulo Oliveira, we try to move on in Parkinson's disease, and we have a study that it's very good to read. When we use some cells from patients for Parkinson's disease, and in fact, with this type of systems, we have good results with this compound. We also have a joint venture with the University of Montpellier, where we are getting results with mice having Alzheimer's disease. And these are the first results, and the compound can decrease lipid peroxidation and decrease the cognitive deficits in showing that it has a protective effect on this system. And also we play in the other disease, ALS, and we have a project that is approved on this topic with Paulo Oliveira, and the first results are coming, and we observe in this system also the protective effect towards several problems of this compound. Then this is the adventures of trying to get funding. We try to play in liver disease. At that moment, Paulo win a project, a European project, a training network, and a RISE project. And we also apply for a national project on this disease, where we try to obtain in vivo data with the same compound, but the disease was the non-alcoholic fat liver disease, and try to show or to get data to evidence that this compound has also protective activity in this disease that is strongly related with mitochondrial dysfunction. In fact, we have good in vivo data, and with the collaboration of the Nenki Institute of Experimental Biology in Poland, we start the first proof of concept in vivo systems, and it was just published at the end of the year, and we realized together that this compound prevents hepatic lipid accumulation by increasing fatty acid oxidation, regulating the control mechanisms, and increasing the antioxidant systems. So, as you see, the goal is always the same, trying to show that these compounds can have protective effect for diseases that are related with oxidative stress. And by now, so far so good, we have some evidence. Meanwhile, you can ask me, and other research, don't try also the same approach? Yes, you know that a lot of research after some concepts and some publications arise, and some research try to put the same carrier in resveratrol, curcumin, quercetin, apo, cinin, some antioxidants that are also in our diet. The question is that they don't produce a small library of compounds. They get one compound and try to check the performance of these compounds. And in fact, they have been very disappointed because the majority of these compounds are very cytotoxic, because we need to modulate the spacer and also the linkage to the antioxidant in order to have a good drug candidate. But as these compounds were very cytotoxic, the research community remember that mitochondrial dysfunction is also related with cancer. And so they move on our endeavor and try to selectively target the cancer mitochondria and try to develop a new therapeutic approach for cancer through disrupting the mitochondrial function. They call the new class the mitokines, and this is an approach that is move on. And fortunately, last year, they also link the same systems to tamoxifen, that is an anti-cancer drug. They obtained the mitotem that is by now in clinical trials, and we hope the best for the success of the application of these concepts in order to induce damage in cancer cells that are metabolically more working than the normal cells. After reading that we can also get good results in cancer fields, we look for the most toxic compound in our library, that is these compounds, and try to see what happened with cancer cells. And in fact, these compounds show a great potential to fight a type of cancer cells by working in mitochondria. So it is a good way to take up some results that we cannot use to prevent this mitochondria dysfunction and see on the other side. And in fact, we ask ourselves why this compound is so cytotoxic, and the others that I presented you before were not cytotoxic. So we can look for this compound. I don't know if you have some chemistry basis, but we can check the right and the left sides. In blue, we have the dietary antioxidant that is chemically a catechol, and we know that catechols can induce oxidative stress. And the other side, we have the famous carrier, the triphenylphosphonate. So we have discussed in the team, and we decide to move and check piece by piece where is the local that gives compound toxicity. This is a work published in the beginning of this year, and it was very pleasant because we tried to look for this challenge, give us the opportunity to look to the carrier that I show you that along this period was maintained intact, and we get start another library, but in this case we replace the phenylphosphonic cation for nitrogen cations, maintain all the chemistry ahead intact. Along this challenge, we need to see if we know that the carrier triphenylphosphonium can enter in mitochondria, but we don't know about the nitrogen cations. And we need to measure if the compounds, to evaluate if compounds can enter in mitochondria. And in fact, we evaluated in rat liver isolated mitochondria, and the compounds with a different carrier can come in also in the mitochondria. And surprisingly, the novel nitrogen, the novel compounds that have the nitrogen carrier were not non-toxic. The concentrations equal and lower than 50 micromolar, and were able to accumulate inside of mitochondria. So we need to check, again with collaboration with Paulo Oliveira, if they affect the main parameters of the respiratory chain, and also some work inside of mitochondria. And we saw that the compound that was cytotoxic based on TPP carrier was can change the mitochondrial membrane potential and decrease the mitochondrial mass. And that these compounds, mainly the carrier based in isoquinoline, we don't have this cytotoxic effect. In fact, some cations show mitochondria parameters similar data, that is a point that can suggest that they can upregulate mitochondrial respiration and increase the threshold for stressing causing damage. So you can ask me, after these adventures, what is the next step? For the next step, that is the plain proof of concept, either of the nitrogen-based carriers and for the developed candidates that seem to work very well, we need, of course, some funding. To translate the knowledge from the academy of the industry, we need a long and quite challenging exercise that is try to find funding in order to finish the proof the concepts and license the patents to the pharmaceutical industry. So we decide to fund a spin-off. We translate this spin-off was funded in 2017 and starts in 2020. Was funded by myself, my group, and Paulo Oliveira and his team, and is dedicated to, of course, target natural antioxidants. They are catching money from investors in order to finish the proof the concept and license the patents to the pharmaceutical industry. Meanwhile, the concept and to finish, the concept was also used for the development of new probes and sensors for targeting mitochondria and evaluating the levels of reactive species. And you see here some examples of probes that are already published. And you see that they have, you see in red, the same cation that we talk along our conversation. So this is the end of my talk. I try to show you that for this disease that have no therapeutic answer, we are facing with complexity, but against complexity, but it is a challenge, a good challenge for the new generation. And I try to show you that we need a team and that together can move the mountains. We need to work very well within the team, but we need collaborations in order that we can attain the goal. Nobody in this field that is very complex must work alone, must share the knowledge in order that we can move on. And thank you for your attention.
Many thanks, Fernanda, for the great talk. Since we have only 1 or 2 minutes left, I will go directly to the questions from our participants.
Okay.
Okay, so the first question is, which would you say is the main obstacle for the delivery of such drugs in vivo?
In this moment, we don't know. We have some in vivo assays, and they work very well. But meanwhile, we have no pharmacokinetics assays that we need to have in order to check the dose if it is correct or not. So we need a lot of data in order to check if they are available. This is the part that I talk that we need more robust data to finish the preclinical studies. We are very far away from the end.
Thank you, Fernanda. Another more specific question, would you say that the systematic understanding of mitochondrial gene function could open new ways for the discovery of novel therapeutics?
Yes, I believe so. But to do that, because in the last years we have a dramatic increase in the knowledge in mitochondria. In fact, during several years in the last century, mitochondria was not looked because they don't understand how it works very well. Till now, we need more knowledge, but this open a new, as mitochondrial dysfunction is common in several diseases, if we can prevent or minimize this dysfunction, and for this we need to have the knowledge of the biologists, biochemistries, and pharmacologists, toxicologists, we can together build molecules that can solve even in precision medicine some problem of orphan disease relates with damage in mitochondria. So it's for me it's a good field to work because it's challenge and I like challenge. And this is for young people. It's a good way to have every day new data.
Great, great. Yeah, this is a multidisciplinary effort anyway. So yeah, another question, are there any databases with mitochondrial disease association data, and can you name a few of them? I don't know if this is your field exactly, but
No, but I check also with Paulo Oliveira in Center of Neuroscience. But till now it is the community. So we work together with Holland and it is not so well organized as the community, scientific community that work in cancer.
yeah.
So we need, I hope, with the new projects in OpenScreen, we need a database that can join together these. It's a good challenge. You, you can see some information in the official sites of orphan disease because they are all related with mitochondrial dysfunction or problem of genetic alteration in mitochondria. And some information is there, but I think that is very, if there is here someone working in databases and so on, it's a good start for a new project to put all this information together.
Great. Okay, another question. Reactive oxygen species kills cancer cells. Why are ROS scavengers toxic to cancer cells? Rather, they should improve their condition. This is a comment from one of our participants.
This new class, I think that is related to this new class of mitochondria. They are not preventing the production or overproduction of reactive oxygen species. They are toxic to mitochondria. It's another look. So they are toxic for the cells. and they can enter in the mitochondria and be toxic to the mitochondria and dysregulate in that sense all the signaling pathways. And the challenge is not to do the same in normal cells. And what we have is that cancer cells are metabolically more active than the normal cells. So the hope is that you can, in some places, dysregulate even more the cancer cells and kill the cancer cells. We are far away from this point. Of course, we need also the help of, I suppose, nanomedicine to develop some systems that can include these compounds and deliver the compounds in the site, in the cells that they are needed. This is opposite from the first part of the history. In the first part, we want to prevent or minimize. With cancer, we want to start a war inside of the cells.
Great. At this point, Fernanda, I would like very much to thank you for your great talk and the great presentation. Thank you so much. I hope we will have the opportunity in the near future to talk more about it. Thank you very
Yes,
much.
I hope so. And thank you for attending. If you need some information, please write to us because we can tell or advise or put you in some collaborations because we need to be all together in order to validate this new road to protect and to find a therapeutic solution for this disease.
This is
Thank
true.
you very much for
Thank you very much.
the invitation.
Thank you. Yeah, at this point, we are close to the end of our webinar. So I would briefly remind you that you can find us online visiting our website, www.eu-OPENSCREEN.eu, or contact us if you have any questions about our training activities in our email address. Of course, you can always subscribe to our newsletter to receive regular updates about our training activities and other services. At this point, I would like to thank you all for being here today and hope to see you soon in our next webinars. Thank you very much.